I was thinking about how incredibly needy the human brain
Oh,
is.
it's the ultimate diva, isn't it?
It really is. It's like this high-maintenance VIP living in the penthouse of our bodies. It demands the absolute best food, it hoards the most oxygen, and it absolutely refuses to compromise on its fuel source.
And that fuel source is overwhelmingly glucose.
Sugar. Exactly. It's frankly a bit of a diva.
It is remarkably demanding, yes.
Yeah.
If you look at the numbers, the brain accounts for, what, only about 2% of your total body weight?
A tiny fraction.
A tiny fraction.
Yeah.
But despite being that small, it devours roughly 20% of your total resting energy. And as you said, it is picky. It wants glucose.
Okay, so that makes sense when we eat. You eat a banana, your brain gets its sugar, everyone's happy. But here's the thing that really blew my mind while reading through our stack for today.
Mm-hmm.
We all know what happens when we eat sugar, but what happens when we don't? Like if I decide to go on a long hike and I run out of granola bars on day 3, or, I'm just sleeping for 10, 12 hours straight.
Or you jump on one of those zero-carb keto trends.
Exactly. My brain doesn't just shut down. It can't.
Thankfully, no. If it did, we wouldn't have survived as a species past the first famine. We'd be in a lot of trouble.
Right. So the question is, when the sugar runs out, how does the VIP in the penthouse stay alive? And the answer, it seems, is basically magic. Or, well, biochemistry.
The body just builds sugar from scratch.
It does. It literally makes it.
It is a biological alchemy called gluconeogenesis.
Gluconeogenesis. It sounds like a spell from Harry Potter, doesn't it? Gluconeogenesis.
Yes, it really does. But it's not magic. It's a very real, very vital process.
And it is literally the difference between life and death for us on a daily, even hourly basis. So today, we are going deep into this process.
We are.
We have a heavy hitter source today. Chapter 19, Gluconeogenesis and the Control of Blood Glucose by Bender and Mays.
This is a foundational text. It's dense, but it's incredibly thorough. It covers the biochemical pathways, the complex regulatory switches that control them, and crucially, the clinical implications when things go wrong.
And our mission today is to unpack all of that. We want to explain exactly how the body manages to reverse a process that is supposed to be irreversible, the breakdown of sugar, and build it back up.
It's like trying to unscramble an egg.
That's a great way to put it. We're going to talk about energy barriers, secret shuttles inside our cells, and why this whole process is so ridiculously expensive energy-wise.
And we should probably emphasize the stakes here early on. This isn't just some abstract chemistry happening in a textbook.
No, this is real life.
Failure of the system is, and the text is very clear on this, usually fatal. If gluconeogenesis stops working while you're fasting, even for a relatively short time, you hit hypoglycemia.
Low blood sugar.
Dangerously low. That leads to brain dysfunction, then coma, and then death.
And on the flip side, what if it works too well?
Well, if it works too well, if the factory just won't shut down, you're looking at the driving force behind hyperglycemia in type 2 diabetes and in critical illness.
So it's a tightrope walk. The body has to keep blood sugar in this incredibly narrow, stable range.
A very precise one. Too little, you die. Too much, you get sick.
Okay, let's start with the basics, the why. We mentioned the brain being the VIP, but is it just the brain that's so demanding?
The brain is the primary consumer, absolutely. The central nervous system as a whole. But there's another crucial player that often gets overlooked.
And that is?
Your erythrocytes or your red blood cells.
The cells that carry our oxygen. I wouldn't have guessed that.
They're absolutely critical. And here's a fascinating distinction between them and the brain. The brain, as we said, prefers glucose. But under extreme starvation, for a prolonged period, it can adapt to use something called ketone bodies.
Right, from fat breakdown.
Exactly. The brain has mitochondria, the powerhouses of the cell, so it can burn alternative fuels if it really, really has to. Red blood cells cannot.
Why not? What's different about them?
They lack mitochondria entirely.
They don't have any.
None. In order to pack as much hemoglobin as possible into that tiny cell to carry oxygen, they've basically stripped out all their internal machinery, including the mitochondria.
It's like a cargo ship that threw all the crew's cabins overboard to make more room for containers.
That's a perfect analogy. And because they have no mitochondria, that means they are entirely dependent on glycolysis, the simple breakdown of glucose, for their energy. They have no plan B.
Wow. So if blood sugar drops to 0, your oxygen transport system literally runs out of gas. That's terrifying.
Precisely. The stakes are incredibly high. So the body has a storage system, of course, to prevent that. We have glycogen.
Sort of sugar.
Yes, it's basically chains of glucose molecules packed together, mostly in the liver and the muscles. But, and this is a big but, how long do you think that supply lasts?
I used to think it was like a camel's hump, good for weeks. But the reading says it's much, much shorter than that.
Much shorter. Liver glycogen is essentially an overnight battery. It's designed to get you through the sleep cycle and maybe into the late morning if you skip breakfast.
So after about 12 to 18 hours of fasting, those reserves are pretty much gone.
They're significantly depleted, yes. By lunchtime, if you haven't eaten breakfast, your body is officially out of its easily accessible stored sugar.
The easy sugar is gone.
It is. And that is the exact moment when gluconeogenesis must kick in. It has to take over the job of supplying glucose to the blood, effectively replacing the role of dietary intake and glycogen breakdown.
Okay, so where is this factory located? If my brain needs the sugar, does the brain make it itself?
No, the brain is a pure consumer, not a producer. The primary factory, the main hub for this whole operation, is the liver.
The liver again. It does everything.
It really is the unsung hero of the body. In the fed state, when you've just eaten, the liver is busy managing all the incoming nutrients. But in the fasting state, it flips 180 degrees and becomes a powerful sugar-producing organ for the rest of the body.
It takes the hit so everyone else can eat. So is it just the liver?
The liver does about 90% of the work, at least initially in an overnight fast. However, the kidneys are also fully capable of gluconeogenesis.
The kidneys, I had no idea.
They are. In a normal fast, they play a pretty minor role. But in cases of prolonged starvation, say several days or weeks, the kidneys can actually ramp up their production to contribute about 40% of the total glucose.
That's huge. I just thought of them as, filters for waste.
They are incredibly versatile organs. The source also mentions there's some debate about the small intestine, that it has the key enzymes, but its overall contribution is probably unclear or very small.
Okay, but there is one place that is conspicuously absent from this list of producers.
The muscles.
Right. And this seems so counterintuitive. Skeletal muscles are full of glycogen, right? They're packed with sugar.
They are. They have a huge private stash, but they cannot release that glucose into the blood to help other tissues.
Why not?
We'll get into the specific enzymatic reason why a bit later, but functionally, you can think of muscles as being selfish.
I like that.
They hoard their sugar for their own needs, for muscle contraction. The liver and kidneys, on the other hand, are altruistic. They make sugar specifically to feed the rest of the body.
Okay, so we know why it happens, to feed the diva brain and the red blood cells. And we know where it happens, the altruistic liver and kidneys. Now let's talk about what we are using as building blocks. If we don't have carbohydrates, what are the raw materials? We can't just make sugar out of thin air.
No, you need carbon skeletons. We call these non-carbohydrate precursors. And the source text identifies a few major ones. The first, and maybe the most elegant, is lactate.
Which comes from exercise, right? That burning sensation in your legs when you sprint.
Yes, that's lactate from anaerobic glycolysis in your muscles. But also, think back to those red blood cells we just mentioned.
The ones with no mitochondria.
Exactly. Since they can't fully burn glucose for energy, they only do the first half of the process, which ends in lactate. They are constantly churning out lactate into the blood, even when you're sitting still.
So the body takes this waste product, lactate, sends it to the liver, and the liver recycles it back into pristine, new glucose.
Precisely. It's an incredibly efficient recycling system called the Cori cycle, which we'll talk more about.
Amazing. Okay, so lactate is one. What's next?
Next up is glycerol. This comes directly from your fat stores. When you need energy, your body breaks down adipose tissue body fat.
Right.
And body fat is stored as triglycerides. A triglyceride is one molecule of glycerol attached to 3 fatty acid chains. The body breaks that apart.
And this is a really, really important distinction the text made. I want to make sure I got this right. The glycerol part of the fat, that small backbone molecule, can become sugar.
Correct.
But the fatty acid part, the long chains that make up like 95% of the mass of the fat, cannot.
That is one of the cardinal rules of human metabolism. It's a key point. Fatty acids are broken down into a 2-carbon molecule called acetyl-CoA.
Right.
In humans and in most animals, we lack the metabolic machinery to turn acetyl-CoA back into pyruvate or glucose. It's a one-way street. So while burning fat provides the energy for gluconeogenesis, the fat molecules themselves do not become the sugar.
That's wild. So you can burn 10 pounds of fat, but the actual carbon atoms from that fat aren't turning into glucose for your brain. They're just providing the electricity to run the sugar factory.
With the small exception of that glycerol backbone, yes, that's a perfect way to put it. The factory runs on fat, but it builds with other things.
Like what? What's the third source?
The third major source is amino acids, specifically what we call glucogenic amino acids
From protein.
from protein. The most important one the body uses is alanine.
And this is coming from muscle breakdown, I assume?
It is. If you're fasting or starving, your body will eventually start breaking down muscle protein to get these amino acids. It ships them to the liver processes them, and uses their carbon skeletons to make new glucose.
So it's a survival trade-off. You sacrifice your biceps to save your brain.
Exactly. Muscle is useful, but the brain is absolutely essential.
There's one other precursor mentioned in a text which seemed a bit odd. Propionate.
Ah, yes. Propionate. This is a huge source of glucose in ruminants, cows, and sheep.
Because of how they digest grass.
Yes, their gut bacteria ferment carbohydrates in their rumen, and a major product is propionate. For them, it's a primary fuel source. In humans, we produce a tiny amount from the breakdown of certain amino acids and what are called odd-chain fatty acids.
So not a big player for us.
Not a big player, but it's biochemically interesting because it enters the whole process in a completely different spot. It comes into the citric acid cycle as a molecule called succinyl-CoA.
We'll probably touch on that later. Okay, so we have our raw materials. Lactate from muscle, glycerol from fat, and amino acids like alanine from protein breakdown. Now we have to build a molecule. And this is where the text gets really intense. It describes gluconeogenesis as essentially being the reversal of glycolysis.
Which is true in terms of the start and end points. Glycolysis takes one molecule of glucose and breaks it down into 2 molecules of pyruvate to get energy.
Right.
Gluconeogenesis takes 2 molecules of pyruvate and builds them back up into 1 molecule of glucose.
But it's not just running the film in reverse, is it? It can't be that simple.
No, absolutely not. And this is all due to the fundamental laws of thermodynamics.
Of course.
In glycolysis, there are 10 steps. 7 of them are what we call near equilibrium reactions. They're reversible. They can easily flow back and forth depending on the concentration of the products and reactants.
So those 7 steps are easy. We can just reverse them.
We can. But 3 of the steps are what we call non-equilibrium reactions. The text called them thermodynamic barriers, which I think is a great image.
I like that too. It's like a waterfall. Water flows down easily, releasing energy. You can't just wish for it to flow back up the cliff.
Precisely. These 3 steps in glycolysis release so much energy that they are, for all intents and purposes, irreversible under the conditions inside a cell. They are one-way streets. So to build glucose, the cell had to evolve clever detours or bypasses around these 3 specific barriers.
Okay, let's walk through these bypasses because this is the real core of the science here. The first barrier is at the very end of glycolysis, or the very beginning of gluconeogenesis, turning pyruvate back into, well, the step above it.
Phosphenolpyruvate, or PEP as it's usually called.
Right.
In glycolysis, the enzyme pyruvate kinase catalyzes the conversion of PEP to pyruvate, and that step generates a molecule of ATP. It's a big energy drop, like going over that waterfall. We can't just push it back up.
So what's the detour? How do we get around it?
It's the most complex of the 3 bypasses. It's a really clever piece of engineering. It actually involves 2 separate enzymes and a trip into and out of the mitochondria.
A trip into the mitochondria, so it starts in a different part of the cell.
Yes. Most of gluconeogenesis happens in the main fluid of the cell, the cytosol. But this first critical step begins inside the mitochondria.
Okay, so pyruvate goes into the mitochondria, and what happens then?
The first enzyme gets to work. It's called pyruvate carboxylase. As the name suggests, it adds a carboxyl group, basically a carbon dioxide molecule to pyruvate.
So it takes the 3-carbon pyruvate and makes it a 4-carbon molecule.
Exactly. It makes a 4-carbon molecule called oxaloacetate. But this process is uphill. It requires energy. It costs 1 molecule of ATP, and it also requires a very specific vitamin cofactor, biotin.
Biotin. I see that in all the hair and nail supplements at the pharmacy.
Yes, but this is its real biochemical job. It acts as a little arm that carries carbon dioxide. It grabs a CO2 molecule and physically sticks it onto the pyruvate.
And this reaction is regulated, I imagine.
Strictly regulated, it requires the presence of acetyl-CoA to be activated.
Why acetyl-CoA? What's the logic there?
Think about what acetyl-CoA signals. High levels of acetyl-CoA mean you're burning a lot of fat.
Right, from the fatty acids we talked about.
So it's a signal to the liver that says, hey, we have plenty of fat for energy. We don't need to burn our precious glucose. In fact, we should probably start making some for the brain. So acetyl-CoA hits the on switch for this first step of making sugar.
That makes perfect sense. It's a fuel sensor. Okay, so now we have oxaloacetate,
Hmm.
but we're stuck inside the mitochondria.
Right, and here's the problem. Oxaloacetate cannot leave. The inner mitochondrial membrane has no transporter for it. It's trapped inside.
So what do? We have a smuggling operation?
We do a molecular smuggling operation. The cell converts the oxaloacetate into a different molecule, malate.
And malate can get out?
Malate does have a specific transporter, so malate crosses the border out of the mitochondria and into the cytosol. And once it's safely outside, another enzyme immediately converts it back into oxaloacetate.
It puts on a disguise to get past security, then takes the disguise off once it's outside the building.
A perfect analogy. It's called the malate aspartate shuttle, but your analogy is much better.
Okay, so now we have oxaloacetate out in the cytosol. We're ready for step 2 of the bypass.
Exactly. The second enzyme takes over, and this one's a mouthful. Phosphenol pyruvate carboxykinase, or PPCK.
DBCK.
I can handle that. PPCK takes that 4-carbon oxaloacetate, and it does two things. It rips off the CO2 we just added.
Which seems wasteful, but okay.
It does, but it's necessary. It rips off the CO2 and uses the energy from another high-energy molecule to phosphorylate it, turning it into P.
Another high-energy molecule,
Yeah.
not ATP.
No, this time it uses GTP, guanosine triphosphate. Energetically, it's the same as an ATP.
So let me just recap this first bypass. To get from pyruvate to PEP, we had to burn an ATP, burn a GTP, use a vitamin, and run a smuggling operation through the mitochondria.
That's it. It just shows how incredibly uphill this process is. In glycolysis, that one step generates energy. To reverse it, we have to pay double.
Okay, that is an incredible amount of work for one step. So bypass 1 is done. We are climbing back up the ladder.
We are. We go through a few of those easy reversible steps until we hit the next big wall.
This is the reversal of the main control point of glycolysis, right? PFK1.
Yep, exactly. Phosphofructokinase 1, or PFK1, is the rate-limiting step of glycolysis. It's the main valve. It turns fructose 6-phosphate into fructose 1.6-bisphosphate.
And that's another one of those thermodynamic waterfalls?
A big one. So to reverse this, we can't just use the same enzyme. We need a specialist, and that enzyme is fructose 1.6-bisphosphatase.
The names are logical, at least. Bisphosphatase means it removes a phosphate.
Yes. It's a hydrolysis reaction. It just uses a water molecule to clip that phosphate off. Very straightforward compared to the first bypass.
So this step is simpler, but it's a crucial regulatory point. The text says the presence of this enzyme effectively determines if a tissue can even do gluconeogenesis.
That's right. It's found in high amounts in the liver and kidney. And interestingly, it's also present in skeletal muscle.
Wait, skeletal muscle kidney? But you said muscle can't release glucose into the blood. I'm confused.
It can't. And that is because of the third and final bypass. This is the key that explains the muscle's selfishness.
Ah, the final gate. The last piece of the puzzle.
The final step in making glucose is to convert glucose 6-phosphate back into free glucose. In glycolysis, the enzyme hexokinase does the reverse, adding a phosphate to trap glucose inside the cell.
So to let the glucose out, we need to remove that phosphate.
We do. And for that, you need the enzyme glucose 6-phosphatase.
And let me guess, muscle doesn't have this one.
You got it. Muscle completely lacks the enzyme glucose 6-phosphatase. So muscle can actually perform most of the steps of gluconeogenesis. It can make glucose 6-phosphate from lactate, for example, but it gets stuck at the very last step.
It can't remove that last phosphate to create free glucose.
Right. And because charged molecules like glucose 6-phosphate can't cross the cell membrane, the sugar is permanently trapped inside the muscle cell.
So it's not a choice. It's a missing exit door. The muscle literally cannot let the sugar out.
Exactly. It's a simple structural reason for a massive physiological difference. The muscle can use that newly made sugar to replenish its own glycogen stores, but it cannot release it into the bloodstream to help the brain.
Only the liver and kidneys have the key to that exit door.
That's right. And that enzyme, glucose 6-phosphatase, is actually located in a specialized compartment within the cell, the endoplasmic reticulum, which adds another layer of complexity to the transport. But the result is clear. Only those tissues can release glucose into the blood and maintain homeostasis for the whole body.
That is absolutely fascinating. Okay, so we've built the molecule. We've bypassed the 3 irreversible steps. But you can't just have this factory running 24/7, right?
Absolutely not.
Especially if you just ate a big meal. If you're eating sugar, you shouldn't be burning a ton of energy to make sugar.
Correct. That would be what's called a futile cycle. You'd be burning sugar and making sugar at the same time, and the net result would be just converting ATP into heat, a massive waste of energy.
So the body needs incredibly tight regulation. The text calls this reciprocal regulation.
Which means, quite simply, when one pathway is on, the other is off. Glycolysis and gluconeogenesis are like two sides of a seesaw.
And the text talks about two different speeds of regulation. There's the slow way and the fast way.
Right. The slow way is what we call induction and repression of enzyme synthesis. This is a long-term adaptation that takes hours or even days.
So this is like if I switch to a high-carb diet for a few weeks.
Your liver will literally start building more of the glycolysis enzymes and fewer of the gluconeogenesis enzymes. It adapts your cellular machinery to your diet. If you fast for several days, the opposite happens.
That's the long-term strategy. But what about right now? I just finished lunch. I need to switch modes in minutes, not days.
For that, you need the fast mechanisms: allosteric modification and covalent modification. These happen in seconds or minutes. They're about flipping switches on the enzymes that are already there.
Okay, let's talk about the master switch. The text puts a huge emphasis on one specific molecule called fructose 2.6-bisphosphate. Now, for everyone listening, don't get this confused with the one we talked about earlier, the 106-bisphosphate. This is 2.6.
A very crucial distinction. Fructose 106-bisphosphate is an intermediate in the pathway. It's a metabolite. Fructose 2.6-bisphosphate is not a fuel, it is a signal, a pure regulatory molecule.
And it's an incredibly powerful signal. The text says it's the most potent positive allosteric activator of glycolysis.
And reciprocally, the most potent inhibitor of gluconeogenesis.
So if the levels of this 2-bil-6 molecule are high, the burn sugar pathway is on, and the make sugar factory is shut down.
Exactly. High 2-bil-6 means we are well-fed, burn the sugar we have. Low 2-bit-6 means we are fasting, shut down sugar burning, and start making it.
So what controls the level of this all-important signal?
This is maybe the most elegant part of the whole system. The level of fructose 2-col-6-bisphosphate is controlled by a single protein called the bifunctional enzyme.
This was my favorite part of the biochemistry. It's one protein that has two different opposing functions, like it has two heads.
Essentially, yes. It's one long polypeptide chain, but it's folded in such a way that it has two distinct active sites. One side has a kinase activity, we call it PFK2, that makes the fructose 2-pol-6-bisphosphate molecule.
And the other side?
The other side has a phosphatase activity, FB-PASE2, that destroys it.
Talk about a conflict of interest. One protein is both the accelerator and the brake for the same signal. So how does the cell know which side to use?
Hormones. Specifically, the ratio of insulin to glucagon in your blood.
Okay, let's play this out. I'm fasting. I haven't eaten in 16 hours. My blood sugar is starting to drop.
Your pancreas senses this and releases the hormone glucagon into your bloodstream.
Right, the fasting hormone.
Glucagon travels to your liver and binds to receptors on the liver cells. This triggers a signaling cascade inside the cell involving a molecule called cyclic AMP, which in turn activates an enzyme called protein kinase A.
Okay, so chain reaction. And what does protein kinase A do?
It phosphorylates things. It finds that bifunctional enzyme and it sticks a phosphate group onto it.
And when that enzyme gets phosphorylated, what happens? Which side wins?
Phosphorylation is the switch. It turns off the kinase side, the part that makes the signal, and turns on the phosphatase side, the part that destroys it.
So as soon as glucagon hits, the liver starts actively destroying the burn sugar signal.
Exactly. The levels of fructose 2-pol-6-bisphosphate plummet. The powerful inhibition on gluconeogenesis is lifted, the activation of glycolysis is removed, and the seesaw tips. The liver shifts into make sugar mode.
That is unbelievably elegant. One little phosphate group added to one protein flips the entire metabolic switch of the liver.
It is. And then, of course, the opposite happens when you eat.
I eat a meal, my blood sugar goes up.
Your pancreas releases insulin. Insulin triggers a different set of enzymes, specifically a phosphatase, that comes along and clips that phosphate group right back off the bifunctional enzyme.
And the switch flips back.
The switch flips back. The kinase side becomes active again. It starts churning out fructose 2-6-bisphosphate. The levels rise, and the liver goes back to burning and storing sugar.
It's amazing how binary it is. It's really like a computer code written in proteins and phosphates.
It is. And of course, we also mentioned acetyl-CoA earlier as well. That's another one of those instant allosteric switches.
Right, the fat burning signal.
Exactly. If you're burning a lot of fat, acetyl-CoA levels rise. That molecule physically binds to pyruvate carboxylase, the first enzyme in gluconeogenesis, and changes its shape to make it more active. It's a direct, instantaneous sensor of what fuel type is available.
We have plenty of fat, fire up the sugar press.
Precisely.
Now, the text also mentions something called substrate cycles, or more ominously, feudal cycles. It said that sometimes these opposing enzymes can be active at the same time. Didn't we just establish that that was a bad, wasteful thing?
It is energetically wasteful, yes. If PFK1, the glycolysis enzyme, and fructose 1-cric-6 bisphosphatase, the gluconeogenesis enzyme, are both working at the same time, all you're doing is turning fructose 6-phosphate into fructose 1,4,6 bisphosphate and back again, and for every turn of that cycle, you burn one molecule of ATP and release it as heat.
So why would the body ever do that? It seems like a design flaw.
Well, the text points out that a small amount of this cycling seems to occur naturally in some tissues, and the theory is that it's not a flaw, it's a feature. It allows for much more sensitive and rapid regulation.
How so?
Think of it like a car at a stoplight. If you have one foot on the brake and one lightly on the gas, the engine is idling, you're burning a little fuel, which is technically wasteful.
Right.
But when the light turns green, you can accelerate much, much faster than if the engine was completely off. By having a little bit of this futile cycling, a very small change in a regulator, like fructose 2-qualose 6-bisphosphate, can produce a massive and almost instantaneous change in the net direction of the pathway.
So it sacrifices a little bit of energy efficiency for a huge gain in control and responsiveness.
Exactly. The body pays a small energy tax for the benefit of being incredibly agile.
That's a great perspective. I never thought of it that way.
Okay, let's zoom out a bit. We've been deep inside the liver cell looking at enzymes and regulators, but the liver interacts with the rest of the body. We touched on this earlier, the idea of recycling. Let's talk more about the Cori cycle.
Right, the lactic acid cycle. This describes the relationship between the muscles and the liver, correct?
Yes, it's a metabolic loop between the two.
So let's say I'm sprinting to catch a bus. My muscles are working so hard they can't get enough oxygen. They're working anaerobically.
Correct. So they are burning glucose very rapidly but incompletely. The end product is lactate.
And that lactate builds up in the muscle, causing that burn.
It does. It then diffuses out of the muscle and into the bloodstream. The liver then picks up that lactate from the blood.
And the liver, being the altruistic organ it is, knows exactly what to do with it.
It uses the pathway we just described, gluconeogenesis, to turn that lactate back into fresh glucose. It then releases that new glucose back into the blood,
Which travels back to the muscle to be burned for energy again. It's a full circle.
full circle.
It almost sounds like a perpetual motion machine.
Not quite, because it comes at a significant energy cost.
There's always a cost.
Always. The liver pays a steep price. To build one molecule of glucose from 2 molecules of lactate, the liver has to spend 6 ATP.
6. That's a lot.
A lot. Meanwhile, the muscle, when it breaks that glucose down to lactate, only gets a net of 2 ATP.
Oof. That is a bad exchange rate. The body as a whole is running a net energy loss of 4 ATP for every single turn of that cycle.
It is. But the purpose of the Cori cycle isn't to create energy, it's to shift the metabolic burden. The muscle is under intense stress. It needs quick, dirty energy right now. The liver is the stable support system. It takes on the energy debt to keep the muscle functioning and to clear out the potentially acidic lactate.
That's remarkably cooperative. It's like the muscle takes out a high-interest payday loan and the liver pays it off later.
That's an excellent way to think about it.
And there's a similar cycle with amino acids, right? The glucose-alanine cycle.
Yes, very similar logic. This is more prominent during fasting.
Yeah.
When muscle starts breaking down its own protein for fuel, it produces nitrogen from the amino acids, which can form ammonia, which is toxic.
Right.
So to get rid of it safely, the muscle combines that nitrogen with pyruvate to form the amino acid alanine. Alanine is harmless. It gets released into the blood.
And it travels to the liver.
It travels to the liver, and the liver is an expert at dealing with nitrogen. It takes the nitrogen group off the alanine, sending it to the urea cycle to be safely disposed of in urine. And what's left over? Pyruvate, which the liver can then turn back into glucose and send back to the muscle or out to the brain.
So it's a way to shuttle toxic waste nitrogen out of the muscle while getting the carbon skeleton back to the liver to be recycled into fuel.
Exactly. It solves 2 problems at once. It transports nitrogen from peripheral tissues to the liver, and it allows for the reuse of the carbon skeletons for energy.
Okay, let's talk numbers for a minute. Blood glucose homeostasis. The body is fighting to keep this in a narrow range. What is that normal range?
In what's called a post-absorptive state, which basically means after an overnight fast, like when you first wake up, normal is between 4.5 and 5.5 millimoles per liter.
And for our American listeners who are used to a different unit, that's roughly 80 to 100 milligrams per deciliter.
Right. After a meal, it can rise, but it should come back down. It might go up to 6.5 or 7.2 millimole, which is around 120, 130 mg/dL. In prolonged starvation, it can drop, but the body defends a floor. It might go down to around 3.3 to 3.9.
And that 3.3 number, that's getting into the danger zone, right? What happens if it goes lower?
It is. Once you drop below roughly 2.5 millimole, which is about 45 mg/dL, you start to see severe symptoms of brain dysfunction, coma. Below that, it's incompatible with life. The body fights incredibly hard with all of these hormonal systems to stay above that 3.5 line.
And the gatekeepers that manage the movement of glucose in and out of cells are the transporters, the GLUTs.
The glucose transporters, yes. They're a family of proteins, and they're numbered. The text highlights a few key ones. GLUT2 is very important. It's found in the liver and in the beta cells of the pancreas.
The cells that make insulin. So what's special about GLUT2?
It has a very high capacity for transport, and it's bidirectional. It lets glucose flow in or out of the cell freely, just following the concentration gradient. This means the liver and the pancreas always have an accurate, real-time reading of what the blood glucose concentration is.
They're the sensors.
They're the primary sensors. Then you have GLUT4.
GLUT4. This is the famous one.
It is. This is the one found in muscle, both skeletal and heart muscle, and in adipose tissue. And its defining feature is that it is insulin-dependent.
This is the locked door we talked about.
It is absolutely a locked door. In the absence of insulin, most of the GLUT4 transporters are just sitting in vesicles inside the cell. The cell is effectively impermeable to glucose.
So the sugar can be right outside, but it can't get in.
It can't. When insulin binds to its receptor on the cell surface, it triggers a signal that tells these vesicles to move to the surface, fuse with the membrane, and insert the GLUT4 transporters. It's like opening the floodgates.
And this just perfectly explains why type 1 diabetics have such high blood sugar. Without insulin, the door in their muscles and fat cells never opens. The cells are starving for glucose while they're literally swimming in an ocean of it.
It's a tragic but biochemically perfect description of the situation.
Speaking of getting sugar into cells, the text makes a big deal about the difference between two enzymes, hexokinase and glucokinase. They both do the same first step, trapping glucose in the cell, but they work very differently.
They do. They both catalyze the same reaction. Glucose plus ATP becomes glucose 6-phosphate. That phosphate group traps it. But their kinetics, you could almost say their personalities, are completely different.
Okay, tell me about hexokinase's personality.
Hexokinase is found in most tissues, like the brain and muscle. It has a very low chemimem, which is a biochemical term for having a very high affinity. It's very sticky.
It's a scavenger.
It's a total scavenger. It can grab glucose from the blood and pull it into the cell, even if blood glucose levels are very low. This ensures that critical tissues like the brain always get fed, but it's strongly inhibited by its own product, glucose 6-phosphate.
So if the cell has enough, it tells the enzyme to stop. A classic feedback loop.
Exactly. It prevents the cell from taking up more sugar than it needs.
And glucokinase, what's its personality?
Glucokinase is found almost exclusively in the liver and the pancreas. Its personality is the opposite. It has a high chemimem, meaning a low affinity. It's not very sticky at all.
So it's lazy.
You could say that. It only really starts working efficiently when glucose levels are high, like after you've eaten a carbohydrate-rich meal. And here's the crucial part. It is not inhibited by its product, glucose 6-phosphate.
So it just doesn't stop.
It just keeps going as long as there's glucose coming in. This allows the liver to act as a glucose buffer for the whole body. When that flood of glucose comes in from your gut after a meal, glucokinase ramps up and traps massive amounts of it, shunting it toward glycogen storage or fat synthesis. It prevents that blood sugar spike from getting dangerously high and overwhelming the rest of the body.
It's the emergency floodgate that only opens during a storm.
A perfect description.
Okay, let's move into the clinical side more directly. We've touched on diabetes, but let's organize our thoughts around the hormones.
Yeah.
We have the two main players, insulin and glucagon, the storage hormone and the mobilization hormone.
The yin and yang of blood sugar. Insulin is secreted when blood sugar is high. Its job is to lower it. It does this by telling muscle and fat to take up glucose via GLUT4. It tells the liver to store glucose as glycogen, and it powerfully shuts down gluconeogenesis.
And glucagon is the opposite.
It's released when blood sugar is low. Its job is to raise it. It travels to the liver and stimulates both glycogenolysis, the breakdown of stored glycogen, and gluconeogenesis, the creation of new glucose.
But the text mentions that glucagon doesn't affect muscle.
That's a key point. Muscle cells lack glucagon receptors. Glucagon's actions are almost entirely confined to the liver.
And there are other hormones too. The text groups them as the diabetogenic hormones.
Yes. This includes growth hormone, ACTH, cortisol, which is a glucocorticoid, and epinephrine, or adrenaline.
These are mostly stress hormones.
They are. And think about it. In a stress situation, fight or flight, or severe illness or injury, what does your body need?
Energy, fuel, ready to go.
And that means high blood sugar to fuel the brain and muscles. So these hormones all act to oppose insulin. They raise blood sugar, largely by stimulating gluconeogenesis in the liver.
This explains why people who are very sick, say in the ICU, often have surprisingly high blood sugar even if they aren't eating much.
Correct. It's called stress hyperglycemia. The body is under such physiological stress that it thinks it's under attack and just continuously dumps fuel into the blood. In critically ill patients, this runaway gluconeogenesis can become a major problem and actually contributes to poor outcomes.
What about the opposite problem, hypoglycemia? The text mentioned this is a particular risk in pregnancy and for newborns.
Yes. Pregnancy is a state of accelerated starvation, metabolically speaking. The fetus is a constant drain on the mother's glucose. It's pulling sugar across the placenta 257, so the mother has to really ramp up her gluconeogenesis to keep her own blood sugar stable, especially overnight.
And the baby after it's born.
Neonates, and especially premature babies, are very vulnerable. They are born with very low-fat stores, so they don't have a lot of fatty acids to power the gluconeogenesis factory.
The electricity we talked about.
Exactly. And on top of that, their own gluconeogenic enzymes in their liver aren't fully developed and active yet.
That sounds like a terrifying combination.
It is. They are highly susceptible to hypoglycemia in the first hours and days of life. That's why frequent feeding is so critical, because their internal sugar-making factory isn't fully online yet.
Okay, let's go back to diabetes mellitus one more time because it ties all of this together. Type 1 versus type 2.
In type 1, the beta cells in the pancreas have been destroyed. There is an absolute deficiency of insulin. Zero insulin is being made.
So the signal to stop making sugar is gone.
Completely gone. The liver thinks the body is perpetually starving because it never gets the insulin signal. So gluconeogenesis runs rampant, completely unchecked. The liver just keeps pumping massive amounts of glucose into blood that is already saturated with sugar.
While the muscles can't take any of it up because there's no insulin for GLUT4.
Which is why the blood sugar levels can go catastrophically high.
In type 2.
Type 2 is different. It's a disease of insulin resistance. The body is making insulin, sometimes a lot of it, but the tissues, muscle, fat, and liver stop listening to it properly. A key feature of type 2 diabetes is what the text calls impaired downregulation of hepatic gluconeogenesis.
Meaning the liver stops listening to insulin's order to shut down the factory.
Exactly. It becomes a leaky faucet that won't turn off, trickling glucose into the blood even when it shouldn't.
And when that blood glucose gets too high, the text gives a number around 10 millimoles per liter.
You hit the renal threshold.
The kidney basically gives up.
Its transporters, the SGLT transporters that reabsorb glucose from the urine, get completely saturated. They're working as fast as they can, but there's just too much glucose coming through. They can't grab it all back fast enough.
So glucose starts spilling into the urine.
Which is called glucosuria. And sugar is osmotically active.
It pulls water with it.
It pulls water with it right into the urine. This causes massive urination, or polyuria, which leads to dehydration, which in turn leads to excessive thirst, or polydipsia. Those are the classic telltale signs of undiagnosed, uncontrolled diabetes.
It all makes sense when you trace it back to the molecular level.
It really does.
Now, I want to get to the final segment, which I think is super relevant to a lot of people today. The energy cost and its link to modern diet trends. We've established that gluconeogenesis is expensive.
Very expensive. To make 1 molecule of glucose from 2 molecules of pyruvate, it costs 4 ATP and 2 GTP. That's the equivalent of 6 high-energy phosphate bonds. It's a huge energy investment.
And where does all this energy come from during a fast?
It comes from one place, fatty acid oxidation, the burning of fat. This is an absolutely crucial link. The gluconeogenesis factory is powered by fat.
So using that information, explain the biochemical rationale behind very low carbohydrate diets like Atkins or keto for weight loss. The text has a specific section on this.
It does. When you restrict carbohydrates to a very low level, say 20 or 30 grams a day, you are forcing your body to rely on gluconeogenesis to supply the mandatory glucose for your brain and red blood cells.
There's no other choice?
There is no other choice. The factory has to run. And because this process, as we've just said, consumes an enormous amount of ATP, The body must burn a significant amount of fatty acids just to provide the power for the factory.
So you're burning fat just to get the energy
Yeah.
to make a little bit of sugar from protein.
That's it. You're creating a situation of high energy demand. This metabolic inefficiency, the high cost of running gluconeogenesis, is one of the key factors that contributes to the weight loss seen on those diets. You're essentially forcing your body to waste energy.
That's a fascinating way to look at it. It's not just about calories in, calories out. It's about forcing a metabolically expensive pathway to run continuously.
That's a huge part of the equation.
So to wrap this all up, we've traced the path from a humble molecule of lactate in a sprinting muscle through the bloodstream to the liver into the mitochondria, through a series of clever and expensive detours that bypass thermodynamic barriers, all regulated by a beautiful symphony of hormones and signals, to end up as a fresh molecule of glucose released into the blood to feed the brain.
It is an absolutely incredible journey against the thermodynamic odds.
And it leaves me with this final thought, this kind of provocative idea. The body is willing to sacrifice incredible amounts of efficiency just for the sake of stability. We burn high-value, energy-dense fats for the sole purpose of maintaining this razor-thin, narrow range of sugar in the blood.
It really highlights the hierarchy of the body.
It does. It shows that the brain is the absolute VIP. The entire metabolic system will essentially bankrupt the rest of the body's energy reserves just to keep the brain online and happy with its preferred fuel.
It will sacrifice muscle tissue, it will burn through fat reserves, all to make a little bit of sugar. The brain's needs are non-negotiable.
So next time you skip a meal or go for a long run, maybe take a moment to appreciate your liver. It's working incredibly hard running this expensive, inefficient, but life-saving factory just for you. Thanks for joining us for this deep dive.
My pleasure.